Hi, everybody. Today, I wanted to just do a quick chalk talk to go through some ideas about antigens and how different kinds of foreign particles are recognized by our bodies. Because we talked a lot about the innate immune system and how the innate immune system uses pattern recognition receptors, right, to recognize if a particle is foreign, if it's a bacteria or a virus or whatever. So now we're getting into antigens. Antigens are any particle that generate antibodies. Antigen, generate antibodies. And what we mean by that is it's able to stimulate an immune response, right? But things can bind to antibodies and be recognized by antibodies and T cell receptors and not stimulate an antigen response. And so there's some definitions we want to talk about first. So an antigen generates antibodies. And I shorten antibodies to ABS. So you'll see that a lot in my writing. Can you see my board? Okay. So antigens generate antibodies. So really an antigen can be anything that a B cell or a T cell can recognize specifically. And we're talking adaptive immunity now. We're looking for a specific response. And so these specific responses are driven by the ability to bind to specific antigens. And that's where we get sort of the recognition of a pathogen that is a specific pathogen. A specific virus has antigens on it that sort of mark it as that virus. And so what can an antigen be? These are usually proteins, but they can be carbohydrates. And they can also be nucleic acids or lipids that are connected to proteins, but usually the nucleic acids or lipids are not in and of themselves enough to be recognized by the structures that make up our T cells and our B cell receptors. But proteins and carbohydrates, right, these are actually pretty complex structures, right? They're not one single thing. They're not simple. They have three-dimensional shape. And so because of that, we can get even more specific when we talk about what it is that our B cells and T cells are recognizing. And so we're going to call that an epitope. Epitope is the actual part of the protein, the part that binds to a receptor. Part of antigen. That binds to receptors. Okay? So, because this is a much more specific thing, right? An antigen can have multiple epitopes. And that's really key when we start talking about how these different antigens affect our immune system and how they're able to be more or less able to stimulate an immune response. So a single antigen, if we talked about a protein, right, is a string of amino acids, right? An epitope might just be this one piece, right? One spot where an antibody can bind, right? And so this one piece, this one protein might have a few different spots where different antibodies could bind, right? And so this one antigen could have many epitopes. Some antigens have only one epitope, some have many. So when we have many, we call them polyvalent. Valent is like where they bind, right? Just like in chemistry. So polyvalent or multivalent means they have more than one epitope. Monovalent means they have only one epitope. So a lot of this is really just vocab, but I want you to get familiar with these terms so that we're not too confused during lecture next week. Okay, so sometimes it's a repeated version of the same epitope within one protein. So it might have repeated sections of the protein. Sometimes there are many different epitopes that are all a separate one. So different antibodies might bind to the same protein in different spots. So those are just all options. That's how epitopes and antigens work together. Hopefully that makes sense. There's a couple of different words I want to talk about. So antigenicity is how good of an antigen a thing is, right? Its ability to bind to B cell or T cell receptors. Just bind. So antigenicity is just binding. there's one step farther. It can bind to a B cell or a T cell, but it may not actually activate that B cell or T cell. And we're going to have a whole lecture on B cell and T cell activation in the future, but I just want you to think about it here in terms of the structural properties of epitopes and antigens. So when we talk about that, we're talking actually about immunogenicity. So immunogenicity is the ability of something to generate an immune response, right? So the words make sense if you break them into pieces. So immunogenicity equals causes an immune response. Okay? So things can be one thing and not the other, right? Things can be antigenic. They can bind to B cells and T cells, but they may not always be immunogenic. They may not actually activate those cells and cause an immune response. And we're going to talk about now what are some of the things that make an antigen immunogenic specifically, make it able to actually cause a whole immune response. And let's see. So some of those things are, so what leads to immunogenicity, right? Maybe I'll just put this down here, is size. So about 10 kilodaltons is the minimum size. Anything less than 10 kilodaltons is too small to be processed and presented to T cells efficiently. So larger antigens are more immunogenic. So larger equals more immunogenic. Okay, so in terms of size, bigger is better if you want an immune response. Some of the other things that come into the immunogenicity are phylogenetic distance. So that means sort of, it's a way of measuring foreignness, if you want to think of that way. So if you imagine our tree of life, right? We have our beautiful branching tree, and it has lots of branches, and we have little branches, right? So if we are here, we are going to have less reaction to an antigen from something here, and more reaction to something from an antigen from over here, right? So the farther away. So bacteria are pretty far away from us on the phylogenetic tree. We react strongly to bacteria. We might react less strongly to an antigen from maybe great apes, for instance, right? Our next closest relative. So that's something to think about, right? We talk mostly about human medicine, but we do use animals often when we're creating biologic medicines. And so we need to think a little bit about how our immune response reacts to foreign things. So the more foreign it is, the more immunogenic it is. Epitope density. So we talked about epitopes and that we can have more than one on a single antigen. If this antigen has a whole bunch of epitopes, it's more likely to be recognized and more likely to cause an immune response. If it has only one epitope, only one spot on that protein where an antibody could bind to, it's going to be less immunogenic, which makes sense, right? So epitope density. The more epitopes, the more immunogenic. And then the last one is degradability. And that seems weird, but we know, and we're going to talk about a lot next week, that this antigen is going to be phagocytosed by a phagocyte. It's going to be broken down into little pieces, and it's going to be loaded on MHC and presented to T cells. If it's made of something that your body can't break into small pieces, it's not going to effectively activate the immune response because it needs to be presented to T cells for the immune response to happen efficiently. And so we're going to talk about all the details of that later, but the ability to degrade an antigen into smaller epitopes does affect whether it can be immunogenic. So there are a couple of funny things that can happen, funny examples. And one of those is called hapten. So a hapten is a very small molecule, a very small protein, usually a protein, that is so small, usually less than 10 kilodaltons, that it cannot be degraded and it cannot be presented. And so even though the hapten can be bound, right, it has good antigenicity, it can be bound by our B cells and T cells, it does not start an immune reaction. And the way we figured that out is really cool. It turns out if you take a whole bunch of these little haptens, I'm going to draw them as little triangles, and you attach them to a carrier protein, then you have overcome the trouble of haptens. So the hapten by itself is too small for our body to recognize, and it will not, well, it can bind to it, but it will not start an immune response. But if you take those haptens and you put a whole bunch of them on a larger protein, all of a sudden it's larger, right? So we got over the size problem. And it's polyvalent because it has multiple binding sites on the same thing. And this can actually cause an immune response. So this putting it on a carrier protein makes it immunogenic. By itself, it cannot start an immune response, but if you put it here, it can. And so what's really neat about this is if you inject just the hapten, you get no response. But if you inject the hapten on a carrier, you get a response against, this is anti, the little a, you get an anti-hapten response as well as an anti-carrier protein response. And some in between, because some might bind in between and bind to both of them. So you get a variety of responses, but you can actually develop antibodies specific for that hapten, but only if you put it on a carrier protein so that it can actually cause the whole immune response, right? You have to make it large and polyvalent. So that's kind of a neat example, and one of the ways that we figured out a lot of this information, which I think is really cool. So I'm going to go ahead and erase this, and we will start talking about some of the other qualities of epitopes specifically. So epitopes, if we remember, are the small parts of the antigen that are actually like bound to a receptor or bound to an antibody, the part that the body actually recognizes on a chemical level, right? That specific lock and key mechanism. So there are a few different types of epitopes. We're going to talk about the three main ones, and we'll see if I can draw them well enough to show this. But the first type we're going to talk about are called conformational epitopes. Conformational. So conformational means it's about its 3D structure, right? It's how it's shaped, conformation. So a conformational epitope is one where the three-dimensional shape of this epitope is part of how it binds. So I'm going to draw a complicated protein structure here, right? This is your amino acids, and it's sort of all twisty-turny folded up into its complex three-dimensional protein. Forgive my artistic talents here. And if you think about it, right, that protein is all folded in three-dimensional space. If an antibody comes and wants to bind to a side of this protein, right, we'll pretend that's our antibody there, the binding part, the part that touches the antibody, the epitope, right, is going to be on multiple sections of that three-dimensional shape. So if you stretch this out, if you denature it, if you boil this protein and you put it with that same antibody, all of a sudden you have this, right, a linear protein, because denaturing it makes it linear. It loses its three-dimensional structure. And these parts where the antibody binds are going to be separate now. They're going to be on this part and this part maybe and over here. And if you put it in with that same antibody, it won't bind anymore because the three-dimensional shape that it bound to is lost. Right? It takes that one little epitope and spreads it across the whole protein because it requires that three-dimensional shape. That's why we call it a conformational epitope. Okay, the next type is a linear epitope. This is probably the easiest to understand. Linear, conformational. So a linear epitope is basically sort of the simplified version of this. If we have our complex protein, maybe it has just one strand on the outside, which happens to be where our antibody binds. So if it binds here, for instance, it's going to bind to this section, right? And that's a linear piece. So that is just a string of amino acids in a row, and it will bind to that string no matter what the three-dimensional shape is, or if it has no three-dimensional shape, right? So if you denature So if you denature this one, it's going to look like a string, but that epitope is preserved because it's still the amino acid sequence is the same, and that antibody will still bind, right? So denaturation does not change a linear epitope. The conformational epitope requires that 3D structure to bind, and when you denature it, that epitope is spread out, it's lost. The antibody can't recognize it. A linear one is the same because it's just a simple amino acid sequence, and it can bind whether it's denatured or not. So the last one is called a neoantigen. Neo means new. And this is one that is, I like to think of this as the antigen is sort of hidden inside the three-dimensional structure of the sort of antigen, the larger antigen. And it's only revealed if it is either denatured or if it's cleaved, right? If there's proteolysis. So in this case, you're going to have this complex three-dimensional structure of a protein, and your antigen, or your epitope, sorry, is way deep in the middle. And who knows, maybe it's three-dimensional too, but let's just call it a linear one that's way deep in the middle of this protein. During normal times, your antibody cannot bind to that epitope because it's hidden in the middle of this three-dimensional structure. But if you cleave it, I'm going to call this proteolysis. Proteolysis is cleaving of a protein, right? Enzymes do this. They'll cut proteins to activate them or whatever. So if this protein gets cleaved, all of a sudden you end up with part of it and another part, right? And your epitope that was in the middle is now exposed on the outside, and your antibody can bind. So this is sort of backwards. The neoantigen version is sort of backwards of conformational. So conformational, the 3D structure is required for the binding of the antibody. If you denature it, that epitope is sort of dispersed and it doesn't work anymore. In this case, the 3D structure is sort of hiding the epitope, and when it is cut, it will show the epitope and the antibody will then be able to bind. So these things are a few definitions and a few basic concepts that I hope that you can take from this lecture, because next time we're going to talk about MHC, which is a major histocompatibility complex, and that's how our bodies present antigen to T cells and B cells. So it's pretty important to understand different kinds of antigens and epitopes and how they work, to understand some of the limits of how our bodies can recognize specific things or can't recognize certain other specific things. And I think this is also really, really relevant if we think about how we design like vaccines, for instance, right? You want to pick an antigen that is as immunogenic as you can get because you want it to generate an immune response and to form memory, right? In the end, that's the goal of vaccination. And so a lot of vaccine development thinks deeply about these structural components. How do you choose the right protein? How do you choose the right conformation of that protein? Do you need to cleave it? What kind of epitopes are there, and are you stimulating the right kinds of cells and the right kinds of antibody production to form protection, right? So these details, they may seem sort of random and a little bit too deep, but they're really, really relevant for a lot of important research that's going on in the medical field, and for development of new kinds of ways of either stimulating because you want to stimulate the immune response, or trying not to. If you're working on pharmaceuticals and some of your pharmaceuticals might have immunogenic properties you don't want, this might give you some clues of ways to get around that or make it more or less immunogenic depending on what the needs are of your future product, whatever it might be. So I hope that's clear. If you have any questions, please let me know, and I will see you all next week. Thank you.